Hello and welcome to a new video on the action potential. So in the last video we looked at how the cell actually generates an electric current, and just to recall, we call this the resting potential, which literally means that the inside of the cell is more negative than the outside of the cell, with the normal difference being about minus 70 millivolts. Now, what I want to look at today is how we can use this biochemical property to send messages from one part of the from one cell to another. Essentially, how we can communicate with our brain cells, and we call this the action potential. And the resting potential that we looked at last time really is the basis for this. So if you don't understand that yet, then please go and look at the previous video first. Just for a quick recall how the resting potential happens, in a nutshell, the difference is in the end that the outside of the cell is positive. So we have a positive charge on the outside, and the inside is negative, and this is caused by several different ions, which basically tend to move for two different reasons. So the ions are affected by both concentrations, these are the two factors that matter, just concentration and charge. Just to put it simply, if there's more ions on one side of the membrane than the others, then they want to find some kind of equilibrium, so they want to have the same amount everywhere. But on top of that, if the inside is negative and the ions are positive, then they will go the positive ones will go to the negative, and the negative ones will go to the positive side. So these factors are responsible for keeping the resting potential alive. And we can also use these same factors now to communicate and send messages. So the third factor that matters, of course, is that they can't just cross the membrane, but they are in fact limited by ion channels. So on top of that, we have ion channels which can be controlled. So in summary, you should remember this from last time, sodium and potassium are the two that really matter. So sodium is mostly on the outside. It would like to come in because the outside is in the end positive, and there's more sodium on the outside, so it has a strong urge to enter the cell, but it can't because normally these channels are closed. On the other hand, the other big ion that matters is potassium, and that is primarily on the inside of the cell. So the concentration would drive the potassium out because there's more on the inside than the outside, but the charge will keep it in because the potassium is itself positively charged, whereas the inside of the cell is now nicely negatively charged. So that more or less keeps an equilibrium, but overall it still has a tendency to drift out slowly, which is why it needs to be constantly pumped back in. If we don't act against this, then it'll basically slowly drift out. Then the other two ions, or the other two chemicals that matter, are a chlorine. So the chlorine, for now, we can sort of ignore. We'll talk about it more when we talk about synapses, but the chlorine is negatively charged. So it's kind of in a similar situation to the potassium. Chlorine is mostly on the outside, and it's quite happy there, because even though the concentration on the outside is bigger than the inside, it has the right charge. So the chlorine itself is negative, the outside is positive, so that kind of keeps it, basically means that it likes to stay where it is. And last but not least, we've got large proteins, and even though proteins are negatively charged, and you can see that there's quite a big difference. So we've got a lot of proteins on the inside and very few on the outside. they actually can't cross the membrane at all because they're quite simply too large. So there's absolutely no way that they're going to go across this membrane. So they stay where they are. But it can be said that the proteins, they are by and large the main reason that the inside of the cell is so negative, because they all have a negative charge and they're going to stay where they are. You can see in the rest of the table the permeability just basically shows how easily these ions can in fact cross the membrane. So as already mentioned before, the potassium can cross it relatively easily, and the other two, the sodium and the chlorine, cannot. And the equilibrium potential down here, the last property, that basically is an indication of at what level this particular ion would be in equilibrium. So the cell normally is in -70, but if it were at +60, that would mean the sodium would not move at all. So basically, if you let these ions move, to what level would they get? So we can sort of see what they would do individually. Right. What we really want to talk about today, though, is the action potential. And the action potential is the way we send messages through the cell. So as you can see here, we see on the graph how charge changes over time. So we're starting off with the resting potential. We're in negative territory here. So as mentioned before, the cell is negatively charged. And to be precise, it is usually -70 millivolts. Slight differences in different parts of the body, but -70 is sort of the standard. Then it goes to a positive charge, and this we call the action potential, and then it goes straight back to a negative charge. So this all happens incredibly fast. You can see on the x-axis, it only takes 4 milliseconds for the entire thing to process. And this really is how our body communicates. So if we put this in computing terms, what's really happening is we go negative, positive, and then back to negative. And really we could say this is one piece of information, one bit. In a computer, bits are zeros and ones, and here they're plus and minuses, but in essence it is the same. So this would be one bit of information. So the smallest piece of information is one action potential. So really we want to look at how our body manages to come up with the action potential, what different factors are involved, and in order to do that we have to look at the different stages of the action potential. You can see here it can be separated into several different stages, how we get to the final action potential. Stage 1 is down here when we are still in the resting potential, but you can see the charge starts to drift towards the positive. Now, why does this happen? So normally we have some sodium that starts to drift in. Why can it drift in? Usually this will be caused by another cell. This could be another nerve cell, or it could be a sensory cell like the eye that responds to light, or the ear that responds to sound. But what this does, it slowly opens some of these sodium channels, and they are actually two different types. So these are the initial sodium channels that are triggered by an external event. So some sodium channels open. As said, triggered by an event. So this event could be another cell. So a synapse actually sending a signal to this cell. So another nerve cell, or it could be a sensory organ. So nerve cell, or a sensory organ like the eye or the ear, or a temperature sensor, etc. So what does that mean? If we open just some of these sodium channels, that means very slowly some sodium will start to drift inside the cell, and it brings in positive charge. So what this means is the cell starts to drift towards the positive, so we go from the initial minus 70 millivolts more towards maybe minus 60, and maybe further towards minus 50. If the signal is strong enough, the charge starts to go towards zero. This is the first stage of the action potential. Now, something important happens when we reach the first trigger point, as we call it, and you can see it here. There's a break in the graph, so this is at minus 50. So if you reach this trigger point here, so I can call this first trigger point, and this is at minus 50 millivolts. This could be called the point of no return. If we reach this point somehow, then basically the action potential can no longer be stopped. So what this means in practice, so this is the point of no return. We now start to see a very strong upshoot, so the charge will now change much quicker, because biochemically something happens, and this something is that we activate a second type of sodium channel. So before we said that some sodium channels are opened by a neighboring cell or so, but here the difference is now that the majority of the sodium channels in our cell are in fact voltage dependent. So these are voltage dependent. And they are triggered to open when the charge of the cell, or not the whole cell necessarily, but just the local area, gets to minus 50 millivolt. Opens when minus 50 millivolt is reached. So that's kind of the magic number here. And now as soon as this happens, quite obviously they're all open at the same time. So now a lot of sodium will enter the cell. So we have basically all of the sodium rushing into the cell, and that means we now get absolutely flooded with positive charge, with the result that the entire cell now flips into the positive, and now we no longer have the situation where the inside is negative and the outside is positive, but in fact it goes opposite, because all the sodium comes in, it brings the charge in, which means the outside goes more negative and the inside goes more positive. So as soon as this happens, basically we have the action potential that will be reached. So action potential reached. So if you go back to our overview graph, we have now reached the point here at the top where the action potential is reached, and as you notice, there is in fact a point where it turns around, and this tends to be at plus 30 millivolts here, because this is where another important point is met. So this is the second trigger point, and from here on it starts to go back down. So the second trigger point. After this, we go into the third phase of the action potential. And the third phase is the repolarization phase. So basically, once it's hit positive, it goes straight back to negative less than a millisecond later. And this drop of charge here back towards the negative is caused by the potassium ions. So if we now look at the third phase, the sodium has done its job. The sodium has entered the cell. but now it's up to the potassium. So we look at the other side. These potassium channels, they are designed to open as soon as we hit these plus 30 millivolts. So this is the potassium channel. And they open when plus 30 millivolt is reached. So now, quite obviously, the potassium that was on the inside before will now start drifting out. Now, we said originally that the potassium is actually quite happy where it is, because even though it has a high concentration in the inside, it doesn't necessarily want to go because the charge is right. Before, when the cell was negative, the positive potassium was quite happy in its place, but now that situation has really changed, because the inside of the cell is now all in the positive, which means the potassium actually has two reasons to leave. So it wants to leave, so basically it starts to move out due to both concentration and charge. Remember saying that these two factors basically kept each other in check initially, but now that situation is quite different. So as soon as the entire cell is now positive, the potassium really wants to go out, and what this does, it sends the positive charge of the cell back to negative, so we actually are back to the original negative charge. Now if you look in a bit more detail, you probably noticed that we even go a little bit lower than we originally were. So normally the resting potential is at minus 70, but we actually enter this what we call the hyperpolarization phase, where it goes a little bit less than minus 70. You can go minus 80, minus 90 millivolts or so. And this is quite important. The reason for this is just that the potassium channels, they're open for a certain amount of time, and they're open a little bit longer than necessary to get to minus 70. So that means that more potassium actually goes out than originally, and therefore the charge goes even lower than in the resting potential. Now this does something important. We will see why this is important later, but basically we call this the refractory phase. So the cell now reacts to this hyperpolarization phase. So we're now in phase number 4, hyperpolarization phase. Hyperpolarization. So this means the cell is something like minus 90 millivolts, and this negative charge has an effect on the sodium channels again. In particular, you can see that this little flap here should demonstrate, we call this the deactivation gate. Deactivation. And that now closes. So what this means is, no matter what happens, the sodium channels can no longer be activated. So even if there is a second action potential following straight up, the sodium channels are in fact deactivated for a little while. So while the charge is so negative, and also basically because it's just gone through an action potential, these sodium channels, they are basically deactivated for a short time. So why does it matter? Well, importantly, we will see in the next video that the action potential actually has to move through the cell, and if the channels get deactivated briefly, that means that they can't get triggered by a second action potential immediately again, and that is important so that the action potential does in fact not move backwards. So if we imagine that the action potential has just been in a certain area, So if we just had it, say, right here, it can only move to areas where it hasn't already passed. So in that case, the action potential can move forwards in the areas that have not just recently been triggered, but it cannot move backwards. So the move in the other direction is not possible because these areas are still deactivated. So it can only move forwards but not backwards. And that essentially ensures that the whole system works in one direction only, that the nerve always passes information from the dendrites down the axon to the synapse and then to the next cell, and it's unable to send this information backwards to where it has already been. Okay, this is really the topic of the next video. So for now, I want to thank you for your attention. And in the next video, we'll look at how exactly the action potential then actually moves through the cell. So I encourage you to look at that one next. Thank you very much.